Hi, I'm Bob Lessig. Welcome to our session today. And what we're going to look at now is a DNA record in the NCBI sequence database. In this case, we are looking at the Homo sapiens breast cancer 1 early onset. That's the BRCA1 gene that's associated with breast cancer risk. It's this transcript variant 1. There are several different transcript variants of this particular gene. But we'll look essentially at the DNA sequence of the BRCA gene and the protein that's associated with it. So this is the actual record. You see Homo sapiens breast cancer 1 is the title. And if we look at the top line, we see that there's an accession number, NM007294. And it's actually a secondary key that identifies this particular sequence. It's 7,224 base pairs in length. It is an mRNA sequence, meaning that this is a spliced gene. The introns have been removed. It's a linear sequence. Really, only bacterial sequences would not be linear. The PRI means this is a primate sequence, so it's in the primate division of GenBank. And it was most recently modified on April 17, 2013. It doesn't necessarily mean when it was submitted. It means that was the last modification of this particular record. On the left, you see locus definition. These are all fields. And in the definition field, you see what's in the title. This is Homo sapiens breast cancer 1 early onset transcript variant 1 mRNA. You see its accession number with a version number. That means this is version 3 of the sequence. So basically, every time the sequence is changed by the author, you will keep the accession number, but you'll get a new version number. So it looks like this sequence was changed twice. The GI or GenBank identifier is a primary key. It's a unique identifier to represent this particular record. If a sequence is changed, the accession number gets a new version, but the GI number changes. And what we're going to notice is a pattern there that suggests that this is a primary key, accession is a secondary key. The keywords field is usually blank or some identifier goes in there. It's an obsolete field, but it's a required field in the database, so it's can't take it away. Its source is the organism it comes from, Homo sapiens. In this case, here is the taxonomic information on Homo sapiens going all the way back to its eukaryotic domain through its phylum and order and class, etc. You see if you publish references relating to this particular sequence, and with the BRCA1 gene, you're going to find a lot. So those references are constantly updated. And you see that there are many references for this particular gene. Often one of the references is the direct submission of the sequence itself. And under the comment field, you see that this is curated by NCBI. You see that on May 19, 2009, this sequence replaced version 632.52871. So you see it's a nuclear phosphoprotein. You get some comments in the comment field about what you're looking at in this particular DNA sequence. Then you see the features table. Features table tells you the source. This is Homo sapiens. The molecule type is mRNA as opposed to genomic DNA, which would be the unspliced version. It's on chromosome 17. We'll see a few synonyms for the genes. Sometimes it's called BRCC1. Some historical names that may no longer be used. Or in some cases, for some genes, there are researchers that call it one thing while another group of researchers call it another thing. So it's useful to have those synonyms there. You see some links to some other databases, and I'll show you some further links shortly. It lists a few of the exons. Exon 1 goes from position 1 to 213. Exon 2 goes from 214 to 312. Most importantly is the CDS region, and CDS is a quite unusual abbreviation. It stands for the coding region of a protein, beginning with the start codon and including the stop codons. And stop codon, even though it doesn't code for an amino acid, is considered part of the coding region of the protein by definition. And you may wonder how coding region becomes CDS. Well, coding sequence. Like I said, it's strange. So CDS stands for coding sequence, and that means that from position 233 in this DNA sequence to position 5824 represents the coding region. Now, the sequence itself is below, and I'm going to click the link that says CDS, and when you click it, it takes you down to the sequence. So here is the DNA sequence itself. you see a series of numbers that suggest this is nucleotide 1, this is nucleotide 61. You see they're laid out in groups of 10. And when I click CDS, it highlighted the coding region of this particular mRNA. So what we're essentially looking at is the start of this mRNA is what's called the 5' untranslated region. It is not translated into protein sequence. The highlighted region represents the CDS or coding sequence, beginning with the start codon, ending with the stop codon. And then after the coding sequence, you'll notice that stop codon is the last 3 nucleotides is TGA, which would be UGA in RNA. And after that comes the 3' untranslated sequence, which is the region of the mRNA after the stop codon that doesn't code for protein. Now, you may notice that there are T's here not used. This is an mRNA sequence. And by convention, U's for uracil are never used in nucleotide sequence databases. So when you see an mRNA sequence, what you're really seeing is the cDNA sequence. The cDNA sequence has the T instead of the U. So note that if this were really mRNA, all the T's would be replaced by U's. The UGA is a stop codon. Going back up to CDS, you see that region, and you also see there's a link to the protein that if I click that, that would take me to the protein sequence for this particular gene, which also was highlighted when I clicked CDS over on the right side of the screen here. And you see the translated protein sequence just in one-letter code for all of the amino acids. And you see some locations of other features of that particular gene. In this case, there are an awful lot of them, more so than the usual record. Now, going down to the sequence, this is called the GenBank format. You see where there are numbers. So if I wanted to find nucleotide 134, I would see, well, this is number 121. I see that from the left. There are groups of 10. This is nucleotide 131. So 134 would be this particular G. It's very easy to find very, very particular positions. However, if I were to copy and paste this sequence, it would have numbers in it. It would be very difficult to do analysis on DNA sequence with those numbers added. So for running algorithms and programs, the most useful format sequence is called FASTA. And if I go to the top of the record and click FASTA, what you will see is a description at the top, and the caret tells me that that's a descriptive line. And below that descriptive line is the actual DNA sequence itself, and that's the G's, A's, T's, and C's that you see here. You see it goes down a long way. If I wanted to get that in plain text, I could do that simply by clicking FASTA text. And now I could select all and run any type of program I would want with that particular DNA sequence. I also want to show you the revision history under Display Settings. If I click Revision History, it will tell me the entire history of this record at NCBI. And if you remember, there were 3 versions of this sequence. So I'm going to scroll way down to the bottom where it will tell me that accession number NM007294, and I should mention that there is an underscore. You can't see it here. The accession number does include the underscore. There's no space in an accession number. This is NM_007294. That's important for search purposes. It was first seen at NCBI on December 9th, 1999. And then the sequence is changed. And usually when the sequence is changed, it usually means they finish sequencing the entire mRNA as opposed to just the coding region. On, it looks like May 11th, 2005. And then the new version was assigned and a new GI number. Gives you an idea of how many sequences are in this database. And it looks like version 2 persisted until May 19th, 2009. And if I wanted to compare what was different in version 3 and version 2, well, I can highlight version 3 and version 2. I'm going to click the left button on the 3 and the right button on the 2. And what will that allow me to do is make the comparison. And I can basically see what changed. It looks like I went from a 7,191 base pair sequence to a 7,224 base pair sequence. And you can see that the pink represents version 3, the gray represents version 2. Okay, now quickly I'll go back to the GenBank version. The default version, I should say, is called GenBank. Now I'm back to the GenBank record. And I want to point out that on the right menu, and they used to be at the top, but unfortunately they've moved them. It's a little hard to find. You see related information. And this will take me to other records at NCBI that are associated with this particular nucleotide sequence. So for instance, if I want the protein that is coded for by this particular nucleotide sequence, I could click protein. And now I have what looks very similar, breast cancer 1 susceptibility, protein isoform 1, except this is no longer the DNA sequence, this is now the protein sequence. I can see that by scrolling down to the sequence and note that there are a lot more than A's, T's, G's, and C's here. There are only 4 nucleotides, but there are 20 amino acids, so I'm using a lot more letters now. Protein sequence is 1,863 amino acids long. But otherwise, the record is set up pretty much in the same way. You see the references, you see the comments, and you see the features table. Now, I'm going to show you a particular region. Now, the BRCA1 protein has what's called a ring finger. Sometimes it's called a ring domain. In this case, they call it a ring finger domain. Sounds like a very interesting domain in proteins. You may wonder where they got the name ring finger. Well, it tells you right here. Ring stands for really interesting new gene, if you can believe that. So here's the ring domain, and it was originally called ring because of a really interesting new gene, but the name stuck. And it's a very particular motif that's found in several different proteins that this one happens to have. It's from region 23 to 68. Now, just like I showed you the CDS in DNA, I could click region, and it will highlight the amino acids that are in that ring domain. And give me a little menu here to describe a little more about what that is. And just like the DNA sequence, I've got a series of links on the right menu. I can get the sequence in FASTA format by clicking FASTA. Here it is, except this is protein and not DNA. And I can get the revision history simply by clicking the display setting for revision history. In this case, there was only one version of the protein. So where the DNA sequence changed, the protein sequence apparently did not. That tells me that the changes were made in the untranslated region, not in the coding region. Or if there were changes made in the coding region, they didn't affect the protein sequence. This sequence was also first seen at NCBI on December 9th, 1999. It's very common that the protein and DNA sequence are submitted together. And all of these changes have been to parts of the record that are not the sequence itself. Meaning that perhaps a reference was added or some minor information was added. We see another whole series of links, and there are a lot of them there, and hopefully as you explore some of NCBI, you'll be able to use some of these links in the future. But that concludes our tour of a sequence record for now. For more information, please contact us in the Center for Biotechnology Information. Thank you.